Welcome to the March edition of the American Journal of Physiology, Heart and Circulatory Physiology, video table of contents, or from the editor's desk. This month, I've highlighted 5 papers that will appeal to a broad spectrum of readers. I know you'll enjoy scanning the entire issue, so let's take a peek inside. Chen et al. Evaluated advanced glycation end product, Ageinduced angiogenesis, in vivo and the involvement of RAGE, ROCK, and moistened phosphorylation in this process. They performed a study in an age-treated mouse model in multiple in vivo and ex vivo models. The results demonstrated that AGEs promote significant neovascularization in whole mount retina and mouse aortic ring of adult and postnatal mice and in a matrigel plug, which were consistently accompanied by increased moistened phosphorylation. The increase of age-evoked neovascularization and moistened phosphorylation were both attenuated by RAGE knockout or ROCK inhibitor Y27632 administration in mice. This study provides in vivo evidence that AGEs induced immature angiogenesis by binding to RAGE, activating the Rho-A-ROCK signal pathway and inducing moistened phosphorylation. Wang et al. Studied cholinergic transdifferentiation in cardiac sympathetic nerves following reperfused myocardial infarction, whereby the sympathetic nerves release both norepinephrine and acetylcholine. They performed MI or sham surgery in wild-type mice and mice in which choline acetyltransferase was deleted from adult noradrenergic neurons. Knockout MI hearts had similar norepinephrine content but reduced acetylcholine content compared to wild-type MI hearts. Knockout MI hearts also had higher basal ex vivo heart rates versus wild-type MI hearts. Action potential duration at 80% repolarization was significantly shorter in the remote and border zones of knockout MI versus wild-type MI hearts, whereas action potential durations were similar in infarct regions. This action potential duration heterogeneity resulted in increased action potential dispersion in the knockout MI versus wild-type MI hearts, which was eliminated with atropine. Mao and co-workers modeled microregurgitation, MR, using simulated echo to evaluate the fundamental assumptions in the echo quantification of primary MR that is due to posterior mitral leaflet prolapse. The theoretical floor of the proximal isovelocity surface area, or PISA, method originates from the assumption that the MR flow rate is the product of the isovelocity surface area and aliasing velocity, which is only valid when the velocity vectors are perpendicular to the isovelocity surface. Other factors such as the Doppler angle effect, the view planes of 2D echo, and the single time instant of PISA were also analyzed. They found that the hemi-elliptic PISA method gives the smallest error for moderate and severe MR cases compared with other PISA methods. Sampil-Vangil et al. Investigated the mechanism by which cigarette smoke affects vascular wall cells and found that cigarette smoke induced a novel form of regulated cell death called ferroptosis in smooth muscle cells. Cigarette smoke extract markedly induced cell death in A7R5 cells and primary rat vascular smooth muscle cells, but not in endothelial cells, which was completely inhibited by specific ferroptosis inhibitors and an iron chelator. Cigarette smoke-induced vascular smooth muscle death was partially inhibited by a GSH precursor, N-acetylcysteine, and an NADPH oxidase inhibitor, but not by inhibitors of pan-caspases, caspase-1, or necroptosis. Duan and colleagues investigated the role of tandem pore domain acid-sensitive potassium, or TASC, channels in cardiac tissue. They determined the role of TASC1 and TASC3 in the pathogenesis of cardiac dysfunction using both human tissue and mouse models of genetic TASC channel loss of function. TASC1 gene expression is reduced in association with either cardiac hypertrophy alone or combined cardiac hypertrophy and heart failure. In a pressure overload cardiomyopathy model, TASC1 global knockout mice have both reduced cardiac hypertrophy, and preserved cardiac function compared with wild-type mice. In contrast to the task 1 knockout mouse pressure overload response, task 3 global knockout mice developed cardiac hypertrophy and a delayed onset of cardiac dysfunction compared with wild-type mice. These findings may provide a rationale for task 1 channel inhibition in the treatment of cardiac dysfunction. Thank you for watching this month's video. Go to our webpage for the entire issue. Have a great entry into spring, and don't forget, send us your best work now.